News release, Wednesday, December 13th, 2023. Genome topology map of human retina development lays foundation for understanding diverse clinical phenotypes in simple and complex eye diseases. Researchers at the National Institutes of Health have mapped the 3D organization of genetic material of key developmental stages of human retinal formation, using intricate models of a retina grown in the lab. The findings lay a foundation for understanding clinical traits in many eye diseases and reveal a highly dynamic process by which the architecture of chromatin, the DNA and proteins that form chromosomes, regulates gene expression. The findings were published in Cell Reports. These results provide insights into the heritable genetic landscape of the developing human retina, especially for the most abundant cell types that are commonly associated with vision impairment in retinal diseases, said the study's lead investigator, Anand Swarup, Ph.D., chief of the Neurobiology, Neurodegeneration, and Repair Laboratory at the National Eye Institute, NEI, part of National Institute of Health. Using deep high-C sequencing, a tool used for studying 3D genome organization, the researchers created a high-resolution map of chromatin in a human retinal organoid at 5 key points in development. Organoids are tissue models grown in a lab and engineered to replicate the function and biology of a specific type of tissue in a living body. Genes, the sequences that code for RNA and proteins, are interspersed throughout long strands of DNA. Those DNA strands get packaged into chromatin fibers, which are spooled around histone proteins, and then repeatedly looped to form highly compact structures that fit into the cell nucleus. All those loops create millions of contact points where genes encounter non-coding DNA sequences, such as superenhancers, promoters, and silencers that regulate gene expression. Long considered junk DNA, these non-coding sequences are now recognized to play a crucial role in controlling which genes get expressed in a cell and when. Studies of chromatin's 3D architecture shed light on how these non-coding regulatory elements exert control even when their location on a DNA strand is remote from the genes they regulate. At each of the 5 key retinal organoid developmental stages, billions of chromatin contact point pairs were sequenced and analyzed. The findings reveal a dynamic picture. Spatial organization of the genome within the nucleus is transformed during retinal development, facilitating expression of specific genes at key time periods. For example, at a stage when immature cells start developing retinal cell characteristics, chromatin contact points shift from a mostly proximal enriched state to add more distal interactions. There also appears to be a hierarchy of compartments that organize the contact point interactions. Some of these compartments, called A and B, are stable, but others swap during development, which further serves to enhance or inhibit gene expression. The data sets resulting from this research serve as a foundation for future investigations into how non-coding sections of the genome are relevant for understanding divergent phenotypes in single gene mutation, Mendelian disorders, as well as complex retinal diseases, Swarup said. The study was funded by the NEI Intramural Research Program, ZAY045 and ZI0546. NEI is part of the National Institutes of Health. This press release describes a basic research finding. Basic research increases our understanding of human behavior and biology, which is foundational to advancing new and better ways to prevent, diagnose, and treat disease. Science is an unpredictable and incremental process. Each research advance builds on past discoveries, often in unexpected ways. Most clinical advances would not be possible without the knowledge of fundamental basic research. To learn more about basic research, visit nui/.dev and news events/basic research media kit. NEI leads the federal government's efforts to eliminate vision loss and improve quality of life through vision research, driving innovation, fostering collaboration, expanding the vision workforce, and educating the public and key stakeholders. NEI supports basic and clinical science programs to develop sight-saving treatments and to broaden opportunities for people with vision impairment. For more information, visit adni.nih.gov. About the National Institutes of Health. National Institute of Health. National Institute of Health, the nation's medical research agency, includes 27 institutes and centers and is a component of the U.S. Department of Health and Human Services. National Institute of Health is the primary federal agency conducting and supporting basic, clinical, and translational medical research, and is investigating the causes, treatments, and cures for both common and rare diseases. For more information about National Institute of Health and its programs, visit www.nih.gov. National Institute of Health, turning discovery into health. QZ BATZ, Sing N, Marchal C, Swoop A, stage-specific dynamic reorganization of genome topology shapes transcriptional neighborhoods in developing human retinal organoids. Published December 2nd, 2023 in Cell, /doi.org/10.016 Cell Rep 2023.113543.